So here, Unit 12, nucleic acids. Okay. Carbohydrates are, when we talk about polymers, those are chains of glucose making up those long polymers. Proteins, those are chains of amino acids, right, making up those proteins. So nucleic acids are what we're going to talk about now here. So there's two types of nucleic acids, what we call deoxyribonucleic acid and ribonucleic acid, DNA versus DNA. We're going to talk about that today. But this is the basis of how we build nucleic acids, right? This is what we can consider a nucleotide. Okay? So again, carbohydrates, right? Those make polymers of starch, glucose. Polymers of starch, amylopectin, glycogen. Then we have amino acids that make up protein chains. And then we have nucleotides that make up nucleic acids. So DNA, RNA are polymers, are chains of nucleotides bonded to each other. Now, every nucleotide has basic commonalities to it, right? Amino acids, it's common to amino acids. There's an amino group, there's a carboxylic group, and then there's some R group, the side chain. So nucleotides will all have some phosphate group. They'll all have this sugar, and they'll all have this base. But the base is what can change, make a different nucleotide, right? The sugar can be modified just a little bit, which I'll show you. So let's first talk about this base, right? There's two types of bases that we're going to see on that particular nucleotide, right? One is considered to be a pyrimidine. That's not the name of the base. That's just the type of the base. Pyrimidine is one ring, right? One cyclic ring that does contain nitrogen, right? And purines contain two cyclic rings, right? So those are two types of bases. Again, that's not the name of the base. Those are just types of base, right? So two bases, purines, two cyclic rings, and then our pyrimidine, one cyclic ring. Now, it's what we attach to them that makes them different. This is where we identify by name, right? Is that here, pyrimidines make up uracil, thymine, and cytosine. Right, so these are the particular names of them, right? So the particular name of that base. And then our purines, there are only two purines that we're going to worry about, right? Adenine and guanine for our nucleic acids, for our nucleotides. But again, we see there's just slight differences here. So between these two purines, here's an amine and it's missing the amine here. Here's an amine group here, but here I have this carbonyl over here. So there's different structures, different groups, branches that these have that make them different. But again, in RNA and DNA, we'll find both guanine and adenine. Right? It's not selective. In DNA and RNA, we'll also see cytosine. But in DNA, we're going to see these four bases: thymine, cytosine, adenine, guanine. And those are the only bases in the nucleotides for DNA. Is that thymine is not found in RNA, and uracil is not found in DNA, right? So these two are specific, right? Thymine is specific to DNA, and uracil specific to RNA. The sugar component in a nucleotide. Both of them are based off of ribose, but there's one difference at carbon number 2. At carbon number 2, this is where we get deoxyribose. So in normal ribose, we see this hydroxy group here. Deoxy is we've removed this oxy, this hydroxide, right? Deoxyribose, and that's the sole difference between DNA and RNA, right? One of them, anyways. One of the differences between DNA and RNA is that this uses a deoxyribose sugar versus ribose sugar, right? And our difference is you can simply just look at the difference here. You look at that sugar. Does it contain the hydroxy group? Does it not contain the hydroxy group? If the hydroxy group is missing, well, then it must be deoxyribose, and we're looking at DNA. We're looking at deoxyribonucleic acid. If I see this hydroxy group there, I know in fact that I'm looking at an RNA structure simply because I see this, right? This is not located in DNA. This is not located in RNA, right? Those sugars are very specific. And then lastly, we have that phosphate group. So it all contains some phosphate group there. So let's talk about building a nucleotide. Right. So first off, there is a nucleoside that's built first. So the nucleoside is our sugar component and one of the bases. Right? Now, even though this is adenine, what type of base is this? There's two classifications, and adenine happens to be which type of base, Gabriella? You can flip back to the page. What type of base is it? Purine, right? That this is a purine here. And I know that because I don't need to know that it's adenine, but I do need to identify it as a purine, and I know that because I see these two cyclohexyl rings, or these two cyclic rings. I keep saying cyclohexyl, but I see these two cyclic rings here, and I can identify that. So once these are bonded here, we get what's called a nucleoside. Right? We get what's called a nucleoside. This is adenosine here in particular. And we're not going to be naming any of these nucleosides. Now, to the nucleoside, right, we add that phosphate group. And this is the structure now of some nucleotide. Right? So here's our nucleotide. Adenosine 5-monophosphate, 5-monophosphate, because this is phosphate. If I added another one here, we would change that to adenosine 5, what? Diphosphate, right? If I added 3 phosphates group, that would be adenosine 5 triphosphate. If you've taken biology, anybody ever heard of ATP? Adenosine triphosphate, right? Difference between, right? AMP, ADP, ATP is we simply just adding these additional phosphate groups onto those structures. And if I'm looking at this structure here, is this a nucleotide? Is this a nucleotide for RNA or DNA? How would I know? So it must be RNA. It's RNA simply because I see this. This is ribose sugar. If it was DNA, sorry, I keep pointing to which one is monophosphate. Yeah, this one is monophosphate here. So this nucleotide here, right? This nucleotide is for RNA. Simply because this is ribose sugar, and I see that OH there. It's not deoxyribose, it's simply just ribose. So our general structure of some nucleotide, right? Some phosphate sugar and then some base. Now this structure here, we're going to have some phosphate bonded to some sugar that's bonded to some base. And we're going to take this nucleotide and bond it to another nucleotide, which also has some phosphate, some sugar, and some base structure. We're going to bond it to another nucleotide with its phosphate, its sugar, and its base structure. And what this creates is what we consider to be that phosphate sugar backbone of RNA and DNA structures, right? So these nucleic acids all have this phosphate sugar backbone, and what makes each RNA and DNA strand different is the sequence of bases. Just like our proteins, the sequence of the amino acids makes every protein different. The sequence of these bases make DNA and RNA different structures. So looking at our primary structure again is just the order of nucleotides, right? For amino acids, primary structure is just, for proteins, the primary structure was just the sequence of amino acids. What order do I put those in? So same thing, for nucleic acids, the primary structure is what's the order of these nucleotides, right? So nucleotides, we say they run 5' to 3' is what we call it, right? And 5' just represents carbon number 5, and 3' simply just represents carbon number 3. And that's how all of these nucleic acids are bonded to each other. Right? If we take a look here, if we're looking at this, this is carbon number 1, 2, 3, 4, 5. So this nucleotide is bonded to this nucleotide from carbons numbers 5 and 3. 1, 2, 3, 4, 5. 1, 2, 3. And there's the phosphate that's bonded between them. 1, 2, 3, 4, 5. 1, 2, 3. And there's that phosphate that bonds those two. So they always run in this 5' to 3' network here when we're looking at nucleic acids. And so the order here running, right, so 5' bond right to 3'. This is the order that we're going to go in now. Is that here's adenosine, cytosine, guanine, thymine. But this portion here, see this portion right here from carbon number 1 to carbon number 5? This, and even including the phosphate, right? So this here is the same structure as this here, is the same structure of this here, is the sugar doesn't ever change, right? The sugar never changes. So when we run 5' to 3', it's much simpler, it's much easier it's much simpler and easier to write it like this versus drawing all of that out, right? If we had to sequence millions and millions and millions of these, we don't want to be drawing these structures. It's much easier just to say, well, I know what the sugar looks like here, I know what the sugar looks like here. The only thing I need to know is the order of those nucleotides. So if we're taking a look at this in particular, is this a nucleic acid for RNA or DNA?
DNA.
What is that? DNA. How do you know this DNA?
Because there's no uracil in it.
Because there's no uracil here, right? We have thymine. What's the other way we can identify this, that this is in fact DNA? Yeah, it doesn't have that hydroxy group. This is deoxyribose. This is deoxyribose, this is thymine, that this is definitely a DNA strand that we have here. You might be, in the homework, you might be asked to, I think in the homework you're going to be asked to draw a structure similar to this. Use this as kind of a template in order to draw that nucleic acid out. But yeah, definitely look at this structure here and just kind of use that as a template to help with that homework problem. These slides are on Canvas, yes. All these slides are on Canvas. Yeah, so for biochemistry, there's so many figures that are large, I can't draw them all out. So I just use slides and then I add them to Canvas. Okay, so we've seen hydrogen bonding. We talked about hydrogen bonding when we were looking at proteins, right? All these hydrogen bonds that are made between the carbonyl and the amino group, is that the bases also interact with each other. Right? The nucleotide here that contains this base here, right? This purine base. This is a pyrimidine base. This is a purine base. This happens to be thymine. This is adenine, cytosine, guanine here. But here is that the bases will form hydrogen bonds with each other. Between adenine and thymine, though, notice that there's only 2 hydrogen bonds here, versus cytosine and guanine, that there's 3 bonds here. This hydrogen bonding is very specific. Is that adenine does not form hydrogen bonds with cytosine or guanine. it will only form bonds with thymine. If it's an RNA structure, it'll form bonds with uracil. Okay? But it's very specific. So cytosine is not going to be forming hydrogen bonds with thymine, uracil, or adenine. Cytosine is going to bond with guanine, guanine with cytosine, adenine with thymine, thymine with adenine, adenine with uracil, uracil with adenine. And it's very specific. Thymine and uracil will never bond because they're completely different nucleic acids. Okay? But the bonding here is very specific. We consider this to be complementary base pairs when we're looking at these structures here. So math that you will have to know for the exam. Right? You don't need a calculator. You should be able to do this in your head. But because these match, because of the complementary base pairing that occurs here, if my nucleic acid contains 30% adenine, and this only bonds with thymine, and none of them are going to be left unbonded, it makes sense that if 30% of my nucleic acid is adenine, then the other 30% must be thymine. So if we take 30% adenine, and the other 30% is thymine, in order to understand 100% of the nucleic acid, how much is left? 40%. So if there's 40% left, how much of it is cytosine? 20%. And the other 20% must be guanine. Right? So it's just very simple math that we can do. Let me change these figures around just a little bit. If I had an RNA structure with uracil, and uracil was, let's say, 20, let's go with 23%. Actually, let's go with 23%. I want to know the percentage of adenine, the percentage of guanine, the percentage of cytosine here. So what's the percentage of adenine here?
23.
It's also 23. This comes up to 46%. How much do I have left?
54%.
54%. How much of that is guanine?
27.
27%, and cytosine must be
27.
27%. Right? So we should be able to, you can bring a calculator if you want, but we should be able to kind of math that out a little bit. But we can definitely find those percentages there. Now, the complementary base pairs, right? We heard the term anti-parallel in Unit 11, right? Anti-parallel means they run the opposite direction, and we have hydrogen bonding that occurs here. So here, if I have this structure here that has, let's see, here's my nucleic acid, and it's running 5' to 3'. Is this RNA or DNA? DNA. How do you know it's DNA? Because it has thymine. Right? We know it's DNA because it has thymine. These make complementary base pairs that run anti-parallel, which means it's going to run 3' to 5' because they're anti-parallel. So adenine always bonds to thymine, and cytosine bonds to guanine, cytosine bonds to guanine, guanine bonds to cytosine, thymine bonds to adenine, and adenine bonds to thymine. As we can get You don't have to draw these hydrogen bonds in, but you do need to know that there are 3 bonds between cytosine and guanine, and there are only 2 bonds between adenine, thymine, adenine, uracil. But it's not necessary to draw those bonds in. Right? Here's our sugar phosphate backbone, right? Our sugar phosphate backbone. But we see that bonding there with those base pairs. And that they run 5' to 3'. Right? So here's our 5' end. This is our 3' end, running anti-parallel 3' end to this 5' end over here. And then we have our phosphate sugar backbone holding those two together. And these will not just form a alpha helix, but actually a double alpha helix, right? Because here we have this backbone here, this phosphate sugar backbone on each strand here, forming what we call a double alpha helix. Running anti-parallel, yeah, I think we've already talked about that, anti-parallel. And again, I'll just note that, right? Running 5' to 3', and then 3' to 5', and we can see all that hydrogen bonding that occurs with that sugar phosphate backbone. And just a little practice here. To make sure we get this complementary base pairing put in. Oh, let's see here. Can somebody read that out to me so I don't have to keep looking? It says starts from A. What's next?
C, T, C, G, T, A.
So, I know it says RNA strand, but let's do what the complementary DNA strand. So our DNA strand is going to run 3' to 5', and we're just going to be doing complementary A'. So A bonds to T, and then this, just read me off of what I write next.
G,
A,
G, C, A, T, T.
And we have our complementary base pair here. Now, if we wanted to run to get the complementary RNA strand, there is a difference for the complementary RNA strand. So we know that this is DNA, and now we want a complementary RNA strand. Knowing that A doesn't bond to T, A bonds to U. So the complementary RNA strand would then be U, G still, right? A, T still bonds to A, G,
C, A, U,
U, right? So adenine bonds to uracil instead of thymine, and this would be some complementary RNA strand, right? So DNA is responsible for the storage, transmission of hereditary information, right? Human cell normally consists of 46 chromosomes. Each one of these chromosomes tightly wrapped around what we consider to be a histidome. And the gene is a segment of DNA that carries out a specific command on how to make other types of cell spores, right? How can I replace a skin cell, right? Grow more hair, make globin, make hemoglobin, right? Make myoglobin, right? And right, 46 chromosomes, all wrapped around some histone. They create these segments here, these genes here. But maybe I don't know. I always appreciate this fact here, is that everything about me, the color of my hair, the color of my eyes, my skin, how tall I'm not, right? All of that comes down to some basic chemistry. Hydrogen bonding between this purine and this pyrimidine base here, and the sequence of those is everything about me. It's just some basic chemistry. all wrapped down here. So inside here we have these 46 chromosomes. And we'll get to that in just, I'm probably going back to this here. But see this little silver button here? Before anybody leaves my class, I'm not really a fan of biology books that, or biochemistry books that show this little silver button. That's not what a histone looks like. A histone is a protein. Right? And we've seen images of proteins. So it's not this histone here isn't this little silver button. It's not a hockey puck looking structure. Is that it's a protein. This colorful thing in here, this is right. This is that structure that we're talking about there, right? This is our histone. This is DNA wrapped around that histone. But it's not, just know that, you know, when you go to biology, take biology and they talk about histones, know that this is what it looks like. It's a protein, right? It's not this silver metal button that we wrap this DNA around. And we're probably going to talk about RNA. I'm going to talk about RNA in just a second. So I'll probably, you know what, I'll just come back to that structure there when we actually get to RNA. So DNA has the ability to make more DNA. Right? They call it replication. Right? DNA can make more DNA simply by creating what we call daughter strands. Right? So here we have A, TAC, So here are two strands. Here's E, this is going to go to A, this is T, this is G, G, and this is T. This isn't the exact process. It's much more complicated. If you want to know more complicated process, we'll take biochemistry or understand it in biology. We just need kind of just a basic understanding at this level here. So we need to know that DNA can replicate and make more DNA. And it does that by complementary basis. So this can go to A, this will go to T, this will go to A, C, C, A, and then over here we have T, A, T, C. Nope, this is G, G, T. And we can make two daughter strands, right? So it's a much more complicated process than this, but we can make new DNA strands. So we know that it can replicate, right? DNA can replicate. Now we'll talk about, introduce the idea of RNA. So major differences between DNA and RNA structures. One, thymine. Thymines in DNA and uracils in RNA. Right? We know the sugar is deoxyribose versus ribose sugar. DNA is a double alpha helix structure, while RNA is just a single alpha helical structure. Right? There's no other half for this RNA structure. And the other thing, you can't see it here, DNA is a very long strand, right? These are quite long, while RNA, not so long, right? RNA is a very small structure, very large structure, very small structure, right? But it's not shown on there. And this is the reason I'm going to go back to the slide here, is that although DNA can replicate itself, right, RNA is duplicated from DNA, and it's kind of like a written set of instructions on how to make stuff, right? Is we get RNA structures that replicate it. They get these directions. Oh, Alex needs more myoglobin. Here's the directions on how to make myoglobin. It sends it out, you know, and our bodies will then, you know, my body will then make more myoglobin for me, right? I get a scratch or something. I need more skin cells. Oh, here's the directions on how to make these skin cells. Go ahead, body, make those new skin cells. These are the directions for it. Okay, so although DNA can replicate, right, this here, this nucleus, right, has what we call nuclear pores. Right? It's just little holes here that chromosomes are too big to pass through. They can't leave the cell because they're too big. But it can make RNA, which is quite small, and those can leave the nucleus. Right? This is how our bodies will get these directions to do all these other things here. So the only three types that we're going to talk about, there's more than just these three, but these are the only three we're going to talk about, is messenger RNA, transfer RNA, and ribosomal RNA. So messenger RNA, right, this is what carries the genetic information from DNA, right, to ribosomes. It's our proteins, our synthesis template to make proteins, right, the directions there. We have transfer RNA that's going to transfer specific amino acids in order to build the protein. And then we have ribosomal RNA. We'll get that to the very end. Ribosomal RNA is more just a structure. It's kind of like a scaffolding used in order to put the protein together. So the central dogma, the central theme, I know it says dogma, just fancy word for theme, okay? The central theme of molecular biology, one we've already talked about, right? DNA has the ability to make more DNA, right? That's kind of this part of the central theme of this, the central dogma, what we call this. So DNA can make more DNA, and through complementary base pairs, we know that DNA also has the ability to make RNA. Okay? So DNA can make more DNA, DNA can make RNA, and RNA makes proteins. Right? So that's kind of the central theme here to molecular biology. So we've already talked about DNA replication. We'll talk about transcription, DNA making RNA. So this is an enzyme here we call RNA polymerase. So this enzyme has one specific job, okay? Is that as we're building these proteins, right? As we're building these proteins, let me get where we're going here. Open. Okay, so we can't unravel our DNA, right? That would cause it to be denatured, right? It's like cooking proteins. We don't want it to become unraveled, but in order for us to make a complementary base pair, we have to be able to open it up. So this polymerase here, this enzyme here has one job, and it's to open a small section up long enough to make this base pair, and then before it leaves, close it back up. Right? Open up just a little bit. We're going to move along. We'll copy and we'll get some base pairing, and then we're going to make sure it's closed so that it doesn't affect our chromosomes and us unraveling altogether, right? So that's this one job here. So we can see that as it's open, we get this complementary base pairing, and again, this is where adenine will bond to uracil, adenine bonding to uracil. We know this is our RNA strand because it's a single, right, alpha helix versus this double alpha helix here. And we enter the state of transcription, right? So initiation sequence where it's opened up, and then the termination sequence here, but running 5' to 3'. But there is an additional step, right? So we have transcription here, but between here and here, this is our synthesized RNA structure. When it's making this, we enter a state that we call post-transcription. So transcription and then post-transcription. Right? We have what's called introns and exons. Right? All of you, I'm assuming, get email, right? You guys have a spam filter, right? So some emails are good, some emails junk, right? Some of these snippets here of RNA are good, some of it's junk. So post-transcription, it's kind of like your spam filter there. So these introns are junk, and these exons all get spliced together. Right? That's the important stuff that we need in order to make these proteins, right? So the introns are cut out in post-transcription, and the exons are spliced together to make that messenger RNA. So now that we have this messenger RNA, we get a sequence. We get this RNA structure that has the sequence of amino acids we need in order to make a particular protein. So let me write this one down here. It can be A, G, so running 5' to 3'. Every 3 bases is considered to be a codon. It's a code, right? Codon, short for code, but every 3 bases is a codon. Right? The codon is a code for one specific amino acid. Right? It's code for one specific amino acid. So we can consider this one here. This one is the first base. This one is the second base. This one is the third base. And then it just repeats itself. This one is the first base. This one is the second base. This one is the third base, right? First, second, third, first, second, third. So every 3 of these 3 bases here is we get some codon, and this codon will represent one of 20 different amino acids. Right? Look at this, right? So here, you'll notice, right? Some of them only have 1. This represents just methionine here, but serine, look, serine has 6 different codons, right? Tryptophan only has 1. Tyrosine only has 2 here, right? So we can see that some have more codons than others. And there's a total of 64 different combinations that we can get for those codons. But again, even though there's 64 codons, they do represent 20. So a total of 64 codons that represent 20 different amino acids. When do we have our exam? You don't have to memorize all. Don't worry, you don't have to memorize. You don't have to memorize this. I'm going to give this to you on the exam. You just need to know how to use this. Okay, so you don't have to memorize it. You just need to know how to use it. Okay, so this is how we use this. It's broken up: first base, second base, third base. So first base, second base, third base. First is A, which here's our A. So somewhere in here is where we're looking. The second base is U. So the second base is U. I'm going to go to this line over here. This is the second base. And now I've narrowed it down to these 4 possibilities. And the last one is G, M-E-T, and this is the reason why we need to know that 3-letter abbreviation. Right? We have to memorize those here because I need to know M-E-T is short for methionine. Right? G-U-C. G. I'm going to go up to U, and I'm going to go across, and here's valine C. So what is the 3-letter abbreviation for AGU? SCR. So A, G, and now I'm going to look somewhere here. I'm going to go to U, and we get SCR, and that's serine. And then we have, so here, let's write this down here, is that we have 5', this was methionine, this was serine, this was, oh no, that wasn't serine. Which one was this one here? It was methionine, GUC, valine. methionine, valine, serine. CCA, what is CCA? 3-letter abbreviation. So this is proline, and what is UAA? UAA is our stop signal. So this signals to stop. That signals to stop, and we would just end it there. That here is our protein with methionine, valine, serine, and proline. Right? So that's how we use this here. So just understand how to use this. Or you could memorize all 64 if you want to memorize all 64 of them by Thursday. Have at it. Or just know how to use this, because again, I will give you that for the exam. Now, with this, no. I want to make sure. I think this was. I don't know if this was added in part of this book or not. I want to make sure it's covered. So it is in there. All right. Your homework. I don't think I saw it in the chapter, but your homework does have it. We talk about mutations that happen. Some random changes that occur when we're synthesizing these proteins. Is something can happen and we have something mutate. So let's say let's say, for instance, this one here. Instead of the codon, this kind of looks a little awkward too. This is not stop. I just wrote stop. This is stop right here, just so there's no confusion. Right. We stopped at the end, and we have these here. So if I'm looking here, what if this mutates? And instead of CCA, it's CCC. Well, what is CCC? Which amino is that? What is it?
Oh.
It's still proline. So if this mutates, it doesn't affect the protein in this case. But let's talk about other cases here. What if this happened to mutate here? What if we had a mutation? Does it change this protein now? So this protein here is now arginine. That can be an issue because this protein might not even work anymore, right? The shape of it's changed, and if the shape changes, the function changes. So this protein that was just made might not be any good for us anymore. Right? So this is what can happen when we do have mutations. Even this is possible to occur here, is that something would happen, and let's say we have right, US or UAC. Well, what is UAC? Right? Well, that means it's not going to stop making the protein. It's going to keep continuing. It's going to make it add more on here until it finally gets to another stop signal. And again, that protein might not be any good, as we don't, make some non-functional protein. So that can happen. We can see these mutations. Some of it affects RNA and these proteins, and some of it does not. Now, so we've talked about messenger RNA. So now we'll introduce transfer RNA and ribosomal RNA. So this big green papasan looking chair here is ribosomal RNA. Right? Think of this more as a scaffolding. Okay? is that it's holding, there's what we call the large subunit and then the small subunit. This is holding the messenger RNA in place, and this skeleton key looking structure, this is our transfer RNA. So it's holding the transfer RNA and the messenger RNA in place until we can form that peptide bond. Right? We form that peptide bond, and then this will just kind of move along. But that's what it's there for. It's there for structure, holding that. And then let's look at transfer RNA. The messenger RNA contains the codon. The transfer RNA contains what we call an anti-codon. So the transfer RNA contains the codon, contains the anti-codon, while the messenger RNA contains the codon. So this codon is specific for some amino acid. I need to move this here. The slide's a little off, but let's take a look at this one here. So ACG is our codon. So if we go to our table, we go to A, C, G, threonine, is that amino acid. So here, ACG is threonine. This is the codon. This is the anticodon. This table here doesn't work for anticodons, right? There's no anticodon table, right? Is that this is for codons, not for anticodons. So the anticodon is on the transfer RNA. So here it meets up, this transfer RNA, and what happens here is that this purple blurple blob here, this is threonine. So this isn't threonine, this is RNA, transfer RNA, and this transfer RNA is specific to bringing threonine. All right? The ribosomal RNA is holding on to the messenger RNA, it's holding on to the transfer RNA, and then along comes CGU. So we'll go over here to our table, and we're going to find C, G, U, and that's arginine. So the anticodon to CGU is GCA because of complementary base pairing, and there it is, and this little red ball here is arginine. So this transfer RNA is next to this transfer RNA, and the ribosomal RNA is going to hold these together until that dehydration occurs, right? And we build that peptide bond in order to form that protein. So the protein isn't formed in here, right? Is that this is not the amino acid, it's the transfer RNA is holding on to that specific amino acid when they're bringing those together. All right. So we've now ended Unit 12.